This is Ethan Russo, presenting for MACE 365. I'm going to share my screen. Our topic today is to entourage or not to entourage. Why is there a question? My email is here. I would be glad to hear of questions. Additionally, any references that I've written from this talk will be available free without copyright problems at ethanrusso.org. Our subject today, of course, is cannabis sativa, cultivated cannabis, the most useful single plant on earth, providing food from the seed, fuel from the seed oil, fiber from the stalks, and pharmacy from the unfertilized female flowering tops primarily. We'll be referring to different typology of cannabis, the system of Ernest Small, type 1 being the most familiar, high THC, chemovars, chemical varieties. There are no strains in plants, rather chemical varieties. Type 2 are mixed THC CBD chemovars, and type 3 are CBD predominant chemovars. This is from an article from a few years ago showing the different parts of the plants and the chemical products derived from them. Of course, the unfertilized female flowering tops are the most popular, with cannabinoid fractions up to 30% on monoterpenoids and sesquiterpenoids that can total 5% exceptionally. These are produced in capitate glandular trichomes, which are most dense in the unfertilized female flowers, quite a bit less in fertilized flowers, on much less on the leaves, although they too can present with useful compounds. And then the roots, which produce triterpenoids and a few alkaloids, quite distinct from the towering tops, but no cannabinoids. We're going to start off with the phytocannabinoids, or plant-based cannabinoids. These are the 12 for which we have the most pharmacological information, although the plant produces upwards of 150 distinct phytocannabinoids. And we'll just be concentrating on a few today, but this is the biosynthetic pathway, generally beginning with a common precursor, geranyl pyrophosphate, which when combined with olivitolic acid produces the pentyl acid cannabinoids, cannabigerolic acid, leading to tetrahydrocannabinolic acid, cannabidiolic acid, cannabichromenic acid. You'll note that these are all acids that's produced in the plant, which after decarboxylation will become the neutral cannabinoids. Geranyl pyrophosphate is also the substrate for the propylcannabinoids. So when combined with divernic acid, produces these with a 3-carbon side chain instead of 5. Additionally, geranyl pyrophosphate is the parent compound to the monoterpenoids and the sesquiterpenoids as well. We're going to be talking quite a bit about cannabis synergy, or what's known as the entourage effect. This was described first in 1998 by professors Meshulam and Ben Shabbat, but the next year they related this not just to endogenous cannabinoids in the body, but also phytocannabinoids from the plant. This type of synergism may play a role in the widely held but not experimentally based view that in some cases plants are better drugs than the natural products isolated from them. This was in 1999. It's now 22 years since that time, and there is a great body of literature that supports this contention, as we'll see. It remains the case that most cannabis chemovars are almost uniformly high THC chemotypes, especially with high myrcene, as we'll discuss. More recently, in the last decade, there's been an emphasis on cannabidiol predominant chemovars, but they're also myrcene dominant. But for decades, people who've used cannabis maintain that there are distinct differences in the effects. So if it's not purely related to the cannabinoid content, what else is it from? Well, that seems to be relative to the terpenoid content and the ratios as we'll describe. Why does the plant make these compounds? Well, THC production is greatest in bright light, particularly in high elevations as in mountains. And this seems to relate to protection from ultraviolet light damage that could affect the genetic contents. CBG and CBD are antifungal. The trichomes form a mechanical insect trap, and we see here trapped aphids. And some of the cannabinoid acids are insecticidal as well as producing a necrosis in plant cells, and that's why they're held away from the plant tissue. And we see an example of pruning as the plant matures. In its effort to get fertilized, it sheds these leaves at the base. So basically then, these are substances that the plant makes not for our use, but rather for ecological benefits. So we'll start with the most familiar, tetrahydrocannabinol or THC. This was finally isolated in 1964. It's a weak partial agonist at the CB1 and CB2 receptors, analogous to the endogenous cannabinoid anandamide. Of course, it's an analgesic and also works on itch. It's bronchodilatory. In the 19th century, there were cannabis cigarettes for asthma. In 1998, Aidan Hampson showed that it's a neuroprotective antioxidant. And beyond receptor activity, it has 20 times the anti-inflammatory power of aspirin and twice that of hydrocortisone. It's a muscle relaxant. That's the basis for its use in spasticity and multiple sclerosis, for which Sativex is approved in 30 countries. Also well known as an antiemetic in chemotherapy. Of course, it's the primary psychoactive component, but it does all these things without producing any COX inhibition, so it does not produce ulcers, heart attacks, or strokes. A very interesting finding is THC as well as CBD may reduce buildup of beta amyloid in Alzheimer's disease. Now we're going to have several slides in cannabidiol, which although it's much less potent and less well known, has a very diverse pharmacology. This is isolated in 1940, but the actual structure was nailed down by Professor Mechoulam in 1963. Does not bind directly to the CB1 receptor, but seems to be a negative allosteric modulator. And this helps account for the ability of it to counteract THC side effects. It also is a neuroprotective antioxidant and is stronger than vitamin C or vitamin E in this regard. Like capsaicin, it's a TRPV1 agonist, but unlike capsaicin, which produces a burning sensation, this does not. But it has the ability to downregulate that receptor with continued application, and this may help explain its role in pain treatment. It also inhibits the uptake of anandamide and weakly inhibits its hydrolysis, so may increase the gain of the endocannabinoid system. It is commonly reported as being sedating, but that is only true at extreme doses, particularly in combination with polypharmacy. In lower doses, it's alerting. The confusion may come about due to the fact that many chemovars of cannabidiol are also rich in myrcene. It's an anticonvulsant and has recently been approved by the American FDA as a treatment for Dravet syndrome, Lennox-Gastaut syndrome, and more recently for tuberous sclerosis as Epidiolex, which is 97% pure CBD. It's also anti-anxiety agent and antipsychotic. There have been 2 phase 2 clinical trials with positive results in treating psychosis at very high doses. Like the other phytocannabinoids, CBD is cytotoxic in breast cancer and many other cancer cell lines, but it has no bad effects on normal cells. It also is an antagonist of GPR18, which may explain benefit in treatment of endometriosis. It's also been shown to be an antagonist of tumor necrosis factor alpha, explaining its benefit in autoimmune diseases such as rheumatoid arthritis, Crohn's, and even multiple sclerosis. Again, not a COX-1 or COX-2 inhibitor. For a long time, it was unclear what the basis was for its anti-anxiety effect, but that seems to be mediated by the serotonin 1A receptor. Also, that's responsible for reduction of brain damage after strokes, treatment of nausea by this agent, as well as other activities. It also enhances adenosine receptor A2A signaling, and that could help explain benefits in inflammatory and chronic pain states. Again, has effects on slow viruses, and also, like THC, can affect beta amyloid production. CBD has also been shown to stimulate bone fracture healing. There are a lot of misconceptions about cannabidiol. As previously mentioned, it's not very potent, so a tiny amount is not enough. So there are many preparations that have almost homeopathic amounts of CBD, and these are unlikely to be beneficial, particularly as isolates. You'll frequently hear that it's not psychoactive, but this is just wrong. Of course, it's psychoactive. What it is not is intoxicating, but it is psychoactive or psychotropic by virtue of its anti-anxiety and antipsychotic effects. We've discussed the sedation business already, related likely to myrcene content. Its status in the United States is a real issue. It is approved and unscheduled as Epidiolex, but technically CBD is still a Schedule I drug in all other applications. Just ask the FDA or the DEA. Now, that has been ignored to date, but we'll have to see what happens in 2021. One of my pet peeves was the contention that CBD turns into THC in the body. This has absolutely been disproven. There was a more recent study out of Brazil where they gave hundreds of milligrams of pure CBD to 2 patients, and no THC appeared subsequently in there as a metabolite. This is the natural history of CBD, all the way from a hemp field to the lab. And we've already discussed the biosynthetic pathway. This is what pure crystals of CBD look like. And as mentioned, it doesn't turn into THC in the body, although that can be done in the lab with strong acid. Rather, its normal metabolite is 7-hydroxycannabidiol, and we're still trying to figure out whether that might be an active drug or not. Cannabigerol is an up-and-comer. This is sort of the parent compound of the other cannabinoids. It is a GABA uptake inhibitor greater than THC or CBD, and may explain its tendencies to be a muscle relaxant and a strong anti-anxiety agent. In animals, it's shown an antidepressant effect. It has a mild hypotensive effect both in the circulation and in the eye. It decreases keratinocytes in psoriasis. It's a powerful antibiotic against multiple resistant Staphylococcus aureus, as well as other gram-positive bugs. There have been various effects on adrenoreceptors and serotonin receptors. It works on several TRP channels, especially TRPM8, which may explain benefit in treating prostatic cancer in particular. Cannabichromene, unlike the others, is produced via a recessive gene and usually is a minor component. It is an anti-inflammatory and analgesic. It shares with the other cannabinoids antibiotic and antifungal effects, similarly sharing cancer cytotoxic effects, and works on various TRP channels as well. Like CBD, it has a certain inhibition of fatty acid aminohydrolase, which is the degradatory enzyme for anandamide, and thus it could boost its levels. Now I'd like to talk about the propylcannabinoids. This is tetrahydrocannabivarin, so it's like THC with a 3-carbon side chain instead of 5, as we see here. This is identified in 1970, but had no work on its pharmacology for a long time until Roger Pertwee picked it up again and showed that it was an antagonist at CB1 at low doses, but an agonist at much higher doses. So a very interesting pharmacology. On various assays in animals and in people, it has produced weight loss, decreased body fat, and serum leptin concentrations with increased energy expenditure. So may turn out to be a useful agent in type 2 diabetes and the metabolic syndrome. It is an anticonvulsant like many of the other cannabinoids. In animals, it's shown decreased edema and hyperalgesia. It lacks the adverse event profiles of the inverse agonists such as rimonabant, SR1417116A, that was marketed as a weight loss drug but taken off of the market in Europe and never approved in the US. Cannabidivarin is its counterpart. So instead of CBD with 5 carbons, we've got a 3-carbon side chain. This is an anticonvulsant that was comparable to phenobarbital and felbamate, particularly for seizures of partial onset. And I should point out that all of the pictures are from dedicated plants rich in these compounds. Now the acid cannabinoids. So the parent before decarboxylation of THC is tetrahydrocannabinolic acid, or THCA, the form in the fresh unheated cannabis flowers. This is insecticidal. It's shown, although it was called inactive for decades, it's far from the case. It is anti-inflammatory via anti-tumor necrosis factor alpha effects, and recently was shown to be a PPAR gamma agonist. The authors of that study in Spain thought it would be useful in Huntington disease, but it also points out that it would be good for metabolic syndrome, cancer treatment, and a bunch of other things. THCA supposedly doesn't cross the blood-brain barrier, but has shown anticonvulsant activity. And the explanation may be that in severe seizure syndromes, there's a breakdown of the blood-brain barrier, and that may allow its entry. This is also a strong antiemetic like other cannabinoids. Cannabidiolic acid is here. This is the predominant phytocannabinoid in fresh hemp. It's a natural herbicide. This one does produce some COX inhibition, but at extremely high doses, it probably never be needed clinically. It is phenomenally powerful in animal experiments as an antiemetic via 5-HT1A stimulation, somewhere between 100 and 1,000 times more potent than CBD itself. Historical data supported the use of raw hemp to treat tumors. This really hasn't been tested subsequently, and that's something that I believe should be done. So terpenoids are the aromatic components of cannabis. Many plants produce terpenoids, commonly from the flowers, fruit, leaf, or sap. They're formed of 5 carbon units. Terpenes have only carbon and hydrogen, whereas terpenoids also may contain oxygen. Monoterpenoids are 10 carbon, limonene and pinene are example. Sesquiterpenoids are 15 carbon, caryophyllene example. One diterpenoid is phytol, which is actually an artifact of heating. And triterpenoids at 30 carbons are found in the roots, but not elsewhere. Again, why does the plant make these? Well, these are insect repellents, or they could be insect attractants. The higher molecular weight compounds in the leaves are antifeedants for grazing animals. They have strong antibiotic properties as well as anti-inflammatory properties. There have been a lot of studies in the past. It's important to understand that the terpenoids are responsible for the scent of cannabis, not the cannabinoids. Over 200 have been reported in cannabis, but about 17 are predominant, and none are unique to cannabis at this point. All the terpenoids that we're going to discuss are generally recognized as safe, grasped by the FDA or FEMA and other bodies. Many of these are food additives. This is a table from the Taming THC article from 2011, and it shows more familiar plants that produce the same terpenoids. And as mentioned, they produce anti-inflammatory, mood-enhancing, or effects, or may modulate the effects of THC. These are monoterpenoids. What they have in common, although they're different structures, is the 10 carbons. And we'll just discuss a few. But by far, myrcene is the most common in cannabis. It's a sedating drug on its own, but particularly in combination with THC, very much so. And it's what I call the couch lock factor in cannabis. This is where people get immobilized and can't get off the couch. But it is useful for other reasons. It's shown to be an analgesic, an effect that's antagonized by naloxone, and this helps explain its sort of narcotic effect, which is very much in evidence in modern chemovars of cannabis, probably to excess. The most common terpenoid in nature is alpha-pinene, but this is actually not seen very often in most available chemovars of cannabis. It is highly bioavailable and is a bronchodilator like THC. Wide-spectrum antibiotic, effective against MRSA, some yeasts, etc. Its most noteworthy effect is an acetylcholinesterase inhibitor. This, by boosting the amount of acetylcholine in the brain, it enhances short-term memory, which is impaired by THC. So this is an important effect to reduce side effects. If you want to wake up, it's good to walk in a pine forest, an effect that the Japanese call Shinrin-yoku, or forest bathing. D-limonene is probably the second most common terpenoid in nature. Again, not that common in most cannabis, but this is a potent antidepressant and immune stimulator, and that was shown in Japan in a study by Komori et al. Looking at a ward of hospitalized depressed patients and cutting their Hamilton depression scales in half and allowing discontinuation of antidepressant regimens. It also works on adenosine A2A, as does CBD and THC. An interesting effect shown recently is it increases mitochondrial biogenesis and can increase the amount of brown fat, the metabolically active fat, and reduce white fat that's associated with obesity. Linalool is familiar as the active agent, if you will, among others, in lavender. So that has a strong anti-anxiety effect that's been shown in hospitalized, agitated Alzheimer patients. It's also a strong local anesthetic and has anticonvulsant effects that have been shown clinically on seizure patients. Sesquiterpenoids, I believe we're only going to discuss one today. These are the 15 carbon examples. Beta-caryophyllene is a fascinating agent. It's been long known that this is a strong anti-inflammatory agent comparable to phenylbutazone, an NSAID that's not used in humans but in horses. But unlike the NSAIDs, this is gastric cytoprotective. About 15 years ago now, Jörg Gertsch in Switzerland showed that this is a selective CB2 full agonist, but has no activity to speak of at CB1. So this is not a psychoactive effect, but rather anti-inflammatory and analgesic without producing intoxication. And it has broad additional pharmacology that can be seen in the review article by Sharma et al. So generally speaking, you hear a lot in relation to cannabis about sativas and indicas. These have no relation to the biochemical attributes of the plant and should be eschewed. Really, we should be defining chemovars on the basis of the chemical content. We're also interested in what the plant looks like, but we really want to know the cannabinoid content and the terpenoid content. Also, what is the scent, taste when vaporized, and what do patients report in relation to its use? So one way to tabulate this would be what's called phytofacts that was developed by Mark Lewis at NAPRO Research. From the top, we see the percentages of cannabinoids and terpenoids. We see a donut with the top 2 cannabinoids, listing of the top cannabinoids by concentration, picture of the bud. Then it gets interesting. What are the primary tastes and smells? So limonene from lemons, caryophyllene from black pepper, as we see here, limonene, caryophyllene. And then very importantly, what do patients report when they use this? Does it produce energy, comfort, less relaxation for this one because of its high, low myrcene content, high limonene and caryophyllene. Other examples, this is a type 2. We've got THC with greater amount of CBD and pinene dominant. So the myrcene's low. This should be a really good drug for treating pain. The pinene will reduce short-term memory impairment from THC, allowing focus and concentration. And with selective breeding, the pinene is very high, 2% out of high total of terpenoids, 3.9%. More commonly, we see things about 1%, but this is a special plant. Again, with selective breeding, without genetic modification, it's possible to have a consistent terpenoid profile with varying cannabinoid content. Here we see a type 1 plant, THC predominant, also pinene dominant. Here we have type 2, CBD and THC, again pinene dominant, just as before. And a type 3, CBD plant with CBG, minimal THC, again pinene dominant. Same thing with limonene. Here, yellow bar is the biggest. We have type 1 here, type 2, and type 3, this time CBD with THC. I should mention that these are not one-offs, chance findings, so these were specifically bred. All the examples here will breed truly. And same idea, this time with caryophyllene predominance. We have THCV and THC together, CBD and THC at type 2, and then very interesting plant, CBD and CBDV, almost no THC, all 3 with caryophyllene predominance. An example of a type 3, CBD predominant with high caryophyllene. So this should be non-sedating. The THC is quite low, myrcene's quite low. What patients report is comfort. This should be good for pain, inflammation, and treatment of addiction due to the CBD and caryophyllene content, as well as having likely anti-anxiety and antidepressant effects. I wanted to point out the importance of flower versus extract. So this is the flower, and you see in this that the terpenoid content is 10.7% of the cannabinoid content, caryophyllene and limonene dominant. So we've got the total cannabinoids at 20.5%, terpenoids at 2.2%. After CO2 extraction, the cannabinoids get boosted way up to 71.6%, but the terpenoids remain quite low, only 2.4%. And this is trying to do things in one run. The caryophyllene is now dominant, and you've lost most of the limonene. So it is better to do CO2 extraction if performed in 2 runs, one for cannabinoids and one for terpenoids. I'd like to talk more about proof of synergy in the entourage effect. We all know that 2 + 2 = 4, but can 2 + 2 = 5? That should be equals, not plus. Well, it can in cannabis. Good example was from the Sativex program. This is a study done in hospice patients with terminal cancer with pain unresponsive to opioids. This was a randomized controlled trial. There were 3 arms, a high THC extract, a placebo, and a high THC extract with a high CBD extract, which is currently known as Sativex or nabiximols. In this study, the high THC extract and placebo were not significantly different one to another, but Sativex was clearly statistically significantly better. And that's demonstrated here. We're looking at a 30% decrement in pain after 2 weeks of treatment. And that's considered highly relevant by the patients. The only difference between the Sativex and the high THC extract was the presence of CBD. So this is an example of synergy. In Israel, Ruth Galili and her team showed an interesting phenomenon. It's known that in animals, cannabidiol as a pure compound produces a biphasic dose response curve. That means at low doses, it doesn't produce analgesia. At a higher dose, which is optimized, it is analgesic, but then the effect is lost if the dose is too high. But when they used equal amounts of CBD as an extract with other components present, the biphasic response was lost, and rather there was a linear dose response effect without a ceiling. So again, this shows the importance of whole plant extracts as opposed to isolates. Similarly, a study done in breast cancer cell lines in culture and implanted tumors showed the superiority of a cannabis extract compared to pure THC. In this instance, the thing that seemed to make a difference was not the terpenoids, but the presence of small amounts of cannabigerol and tetrahydrocannabinolic acid acting as synergistic agents. And all these examples are detailed in a publication from a couple of years ago now, which is listed here and available at ethanrusso.org. Another study done out of Israel and Canada, looking at anticonvulsant effects in an animal model with pentylene tetrazole of different cannabis extracts with equal CBD concentrations. All the extracts showed some benefit, but the big differences were noted with non-CBD cannabinoids, leading to very significant differences in survival rates in the experimental animals. So these so-called minor components may be the difference between clinical success and failure. Also demonstrating this was a meta-analysis by Fabrizio Pamplona et al. And they looked at studies of pure CBD as an Epidiolex in treating severe seizure syndromes as compared to similar dosing, but with extracts with other components. What they showed was that purified CBD, the average dose per day was 27.1 milligrams per kilogram, compared to equal success with only 6.1 milligrams per kilogram per day for the CBD-rich cannabis extracts. So that's about a 22.5% of the higher dose. Also, the adverse event profile was improved in the extracts over the high dose CBD alone. And this is important because at high doses, there are drug-drug interactions of CBD with clobazam and other agents, which may complicate treatment. Also, adding markedly to the expense. It should be known that other components are anticonvulsant beyond CBD, including THCA, THCV, CBDV, possibly CBDA, linalool, and even caryophyllene. Good example of that is this type 3 agent, CBD with CBDV. Also contains a small amount of THC and linalool and caryophyllene, so at least 4 potential anticonvulsants. This type of chemovar should be an ideal candidate for testing clinically for epilepsy. Some other examples from the realm of psychiatry. Depression, one might think about THC, CBD, and CBG plus limonene. For anxiety, CBD plus limonene and or linalool. For sedation, THC, less CBN. CBN, cannabinol, is about 1/4 of the potency of THC, but especially with myrcene. For agitation and Alzheimer's disease, THC, possibly CBD, along with linalool, limonene, and pinene. And for addiction, as mentioned, CBD plus caryophyllene, and maybe a touch of THC. I wanted to close with something about biosynthesis of cannabinoids by yeast and other methods. This was from a study of a couple of years ago, and this showed at the time what yeasts and other species have been used via genetic modification to produce cannabinoids. 2 weeks after this came out, a more elegant study was done that included a lot more agents. But the point I wanted to make was the amounts produced were tiny. Industry says that they can scale this up, but I would point out that they might be good at producing isolates, but they'll never produce the entourage that cannabis does. And it's hard for me to believe that they can compete with a plant that can produce 30% cannabinoids in the flowers and 5% of the terpenoids. So, there are a lot of neat tricks that can be done with genetics, such as producing anole lizards that are albinos, but this may have a certain appeal to people, but ecologically, it's not good for the animal. And I just wanted to close with something about land races. Land races are chemovars of cannabis that have been grown outdoors in a geographic location, so they're ecologically adapted to the local conditions. And you'd find these in traditional cannabis growing regions such as Mexico, Southern Africa, Jamaica, Colombia, Morocco, Afghanistan, India, Thailand, Nepal. Unfortunately, in those places, a lot of the land races have been supplanted by seeds from California and the Netherlands that really don't necessarily grow there well. In the process, we've lost many wonderful traits, profiles, as well as agronomic benefits, disease resistance, etc. We definitely need to make efforts to find these plants and preserve their genetic legacy. And I believe that's it. I thank you for your attention. And again, I'm available at Russo@comcast.net. Thank you.